Okay, Dr. Philip Motta is the professor of biology at the University of South Florida. He got his Bachelor of Science degree at Duke University and his PhD at the University of Hawaii. He is the past president of the American Elasmobranch Society, and he works on the study of the functional morphology of sharks and bony fishes, and that's what he will be talking with us today about. Is the functional morphology, feeding anatomy, you can see, read the whole thing. Filtering rate and diet of whale sharks off the Yucatan Peninsula in Mexico, and that part of the research is being funded and sponsored by the Georgia Aquarium. So we're really anxious to hear what you have to say. So Phil, take it away.
Thanks. Can we dim the lights a little bit? You did already, okay. Well, it's hard to see with the authors here, but I just wanted to point out some of the names that maybe you can't see. Mike Maslanka, who was here before, Bob Huter from Moat, Ray Davis, who used to be here, Rafa de la Parra from Mexico, and a bunch of my grad students, Samantha Mulvaney, Maria Habiger, James Struthers, I'll tell you about a little later on. He's a fluid dynamics person. And Kai O'Mara, Jane Gardner, John Tominski, he used to be at Moat, and Leslie Ziegler. So, let me tell you a little bit about what we are doing with these guys, if I can get it to go to the next one. Takes a little while. 1 second. There. Okay. Whoop, go back already. This is work sponsored by both Moat Marine Lab and the Georgia Aquarium, and it is conducted at, as we'll see in a second if it comes back to it, off the Yucatan Peninsula, right off here. And we are working with probably the largest aggregation of whale sharks in the world, it seems to be. On some days there may be 140 sharks on the surface out there. Right off, if you recognize, Cancun is down here. We're in the Yucatan Peninsula up here, and the whale sharks are generally off of there. It's a protected area by the Mexican government, and ecotourism is a big, really big deal down there. So the research has nicely integrated with ecotourism in this area. This is what it might look like on a typical day in the summer, towards the end of June, July, August, around there. And you'll have numerous whale sharks feeding on the surface like that, and you can see a tourist boat in the background. Interestingly, the tourist boats have very strict regulations about number of people in the water with the whale sharks, no touching of the whale sharks, no riding the whale sharks, and so on. So the Mexican government is actually very much on the ball with this. We actually run into a little problem when we are doing research because the locals complain, the guides complain when we have too many people in the water. They say we're breaking the rules. So we have to be very careful. So the work is done, of course, in Holbox. It's pronounced Holbox, Mexico, the Georgia Aquarium. I was just doing some a little while ago, about half an hour ago. We use surface and underwater video photography. We take gait measurements. I'll show you that in a while. We were doing some of that today. Filter pad anatomy, plankton toes, nutrient analysis with you guys here, and computational fluid dynamics will come in at the end. So basically, what you guys are probably seeing here is some of the different feeding behaviors of whale sharks, the different ways. And the names are, these are some of the names that previous researchers actually given to it. It's called active surface ram filter feeding, which is when the shark, as you see here, swims on the surface, right on the surface like that. And it's ram feeding means when the shark swims forward and rams the water through its mouth. And it's feeding on plankton on the surface. Then there's pulsatile subsurface suction filter feeding, which is shown there on the right. Occurs in many places, including some places off Mexico. The animal turns not completely horizontal, sometimes more than others, and uses a pulsatile suction behavior to suck the plankton in. And you can actually see the vortices being formed, being sucked down, with seaweed being sucked down into the mouth. Quite powerful. And then you have the next level down is passive subsurface ram filter feeding, where the animals go below the surface like this and they swim with the mouth open to filter feed. And I'll come back to these in a while. These are pictures taken by our group and some of the Mexicans working with us. This is going to be used in a publication that we're working on right now. And this is on this talk, I'm talking about the active ram surface filter feeding only. This is the behavior we recognize easily. We can document it on the surface. The visibility is very low because the plankton is so rich there. So working under the surface is not very easy. And when we see animals subsurface swimming off Holbox, they seem to have the mouth closed. We think, for the most part, they're feeding on the surface. And this is what it looks like from the side, as you've seen here from the when they're feeding somewhat. They seem to be at an angle of, my estimate angle is 26 degrees, and that's what Al's going to try and look into and see if we can measure it here. I want to see what that angle is that they're plowing through the water. So some of the mouth is out of the water. And what we do is we follow it in boats off Holbox these are the Mexican park authorities. So we work with them. And we're using a technique that I think you use as well. We have dual green laser beams calibrated 50 centimeters apart. And the lasers are shining on the head of the shark, which gives us an absolute distance measure. And we're filming with a camcorder. This is the camcorder that actually does high-speed video. It shoots up to 240 frames per second. We're normally shooting at about 125 frames per second. So what we're doing in many cases is we're looking at we track it with the boat, side beside it with the boat to get the swim velocity when it's feeding. We do that. We take observational, we take video as well, and then we actually time the flow rate of particles into the mouth because we want to calibrate. We're running with the boat and the GPS. We want to know what the speed, the filter feeding speed is, but we want to make sure it's correct. So we check it with timing particles going into the mouth, and what we did is we tried all different kind of things because sometimes particles are not in the water. So I always go to the simplest possible thing. We took instant rice. I soaked instant rice in seawater, and I would throw instant rice in front of the sharks. And if it did, in most cases they would dive right under it. I squirted food coloring in front of them, and they'd dive right under the food coloring and come back up every time. They see it. We actually tested this because we put salt water back in the syringe and squirted in front of them, and they don't dive. It's only when there's a color in front of them they dive. In some cases, the rice does go into the mouth and actually spit out. These are about 25-foot animals. A handful of rice like this much, they spit it right back out immediately. And then we found that the simplest thing again, quite often the simplest thing is sargassum. We picked up seaweed, we threw it in front of them, and we timed the rate at which the sargassum went into the mouth. Okay, Mike and you guys from your crew from here, at the same time, when we finished sampling a shark, you would run right behind the shark where the shark was feeding, and we would do a plankton tow, a very timed, calibrated plankton tow to get the volume of plankton and the species of plankton, etc., that they're feeding on. We'll come back to that in a while. So what I need to know, and that's what we're trying to do this morning, and we're trying to get measurements from you as well, is I need to know the dimensions of the open mouth. Here's the shark plowing through the water, and I'll show you in a while, part of the mouth is above the surface like this, in most cases. So I took 128 measures using photography, both video and digital photography of the sharks coming straight on like this, and I can get the relative area of the mouth that is out and the relative area that is below. So I do a mean. I do numerous sharks and about 128 observations that way. So what I do is I also, in some cases when the shark is coming straight at us, it's very tricky, but I can shine a laser beam right on their nose. And so I know the laser beam is 50 centimeters apart, so now I know the size of the head, the mouth. I actually have measurements of the mouth diameter, so I can get from that, I can get the actual size of the mouth, because it's a very convoluted way. I know, that's what we were measuring this morning, the distance between nares. I know the ratio of the distance between the nares to the mouth height and the mouth width and to the length of the shark. So based on this interneural distance, if I can get interneural distance, I'm pretty accurate and tell you the size of the shark. So all these measurements give me a mouth size. I get a mouth size, and I use photographs like this of sharks filter feeding, and as I've seen a while, I will then, knowing the size of the shark, I can get the size of the mouth. Knowing the size of the mouth, I calibrate this. Let's say this is 56 centimeters. I calibrate it in my computer program. I block in this area here, the internal width of the mouth, and now I have the area of the mouth plowing through the water. See? So that's why I need to measure the animals that are here. It's so important to do that. So you can see some of the ratios there on the bottom. There's very distinct ratios of mouth width, the height, length, and so on. So what do they swim at? They swim, the mean is 1.1 meters per second. You can see the standard error and the range and so on. This is actually very, very similar. This is when they're filter feeding. Very similar to what they see in Australia and off Belize. It's very constant. The percent of mouth above the water for about 128 measures is 15.3% of the mouth is above water. It varies from 0 to 20-something, but basically 80-something% of the mouth is, or 84% or so, is below the water. So we have to take that into account. We can't take the area into account above the water. So, all right, we sidetrack for a second and look at diet. So we collected the plankton from down there off Holbox, And we did nutrient analysis with you guys here and also with Mike Maslanka, who is now at Smithsonian. So what are they feeding off in Holbox? Well, in the feeding areas where they're feeding, they're using shrimp. Most of the thing is shrimp, little suggested shrimp, which are small planktonic shrimp, calanoid copepods, chaetognathes, and fish larvae. And fish larvae are very small, by the way. We'll come back to that. In the non-feeding areas, we do controls. We run over to our control area, and we sample in the control area, and they're feeding on not in the control area. It's basically the same thing, except you notice that shrimp are dominating. They're pink in color, so the water looks blood red where they're feeding. Now, if you compare that to Ningaloo Reef in Australia, they're feeding on crab larvae, stomatopods, krill, etc. La Paz, Mexico, by Clark and Nelson in a study, calanoid copepods, earlier study. In Baja California, Nelson and Eckert, they found calanoid copepod shrimp, crab larvae, etc. Belize Barrier Reef, there's a place off there where they eat fish eggs almost exclusively off Belize Barrier Reef. So, in general, based on all the studies, for the most part, the whale sharks are planktonic feeding, filter feeding animals. You'll see in the literature that they eat tunas, they eat jacks, they eat bait fish, all that stuff, and I think a lot of that is incidental. The bait fish swim with the shark right beside the mouth, continuously with it beside the mouth, and what I think people mistake sometimes is the bait fish going into the mouth. Although sometimes they will do a behavior where they'll come out of the water and they'll sink down in the water, and in some cases I think some bait fish do get trapped in there, and they probably can swallow them. But they're not targeting tuna and big fish, I don't think. All the evidence point towards plankton primarily, zooplankton. So we then meticulously, I don't do this, my grad students do it, that's why I have students. They sit down and measure hundreds and hundreds and hundreds of the little plankton. We sort them out, we get subsamples, and we measure their length and width, because we want to see at the feeding site if the size of the plankton differs from that of the non-feeding site. And it does for some of them. For instance, for shrimp, you can see that the length and the width of the shrimp is actually significantly greater in the feeding places versus the non-feeding. These could be a couple kilometers apart. I mean, we're talking an area which adjacent to it could be a non-feeding area, and there's a significant difference. And you'll see like arrowworms are slightly greater in width in the feeding area, and copepods are actually slightly bigger in the feeding areas than the non-feeding areas. So there is a difference in plankton. Now look at some of this. Look at fish eggs. Pay attention to this. Fish eggs, this is in millimeters. Fish eggs are about 1 millimeter. About 1 millimeter in diameter. We'll come back to this in a while. So what about prey density? Well, from these data, we can also get prey density. And what we find is in the active ram filter feeding, that stuff on the surface there, the patches have about 2,000 plankters per cubic meter in these feeding areas. If you look at the background areas and non-feeding areas where these guys are, it's significantly less, the density of plankters. So there's far more plankton in the feeding. They are selecting plankton-rich areas. This has been shown for basking sharks and for whale sharks. They are selectively picking plankton-rich areas. We don't know how. Probably smell, not sure. Interestingly, Baja California, Nelson and Eckert study, beautiful study. They found, look at the difference in the density of their plankters there. 86,000 plankters per cubic meter. They found very interesting, when the plankton density was the highest, they did that feeding. When it became lower, the plankton density, they switched to that feeding. And when the plankton density went to the lowest density, they went below the surface and started doing that feeding, which we can't verify that at our site because all we're observing is the surface feeding. So they seem to switch depending on the density of the plankton. All right, nutrient analysis again, which we have done through you and through the Smithsonian. If you take a batch of this plankton, a whole big batch of it, and analyze it, we find out that it's of course, we're doing wet weight. So water is the primary component of it in terms of wet weight. And there's protein, the lipid levels, and the ash content, and so on, and the energy. And what really concerns us at this point is the energy is 0.324 kilocalories per gram of wet weight, or 0.292 kilocalories per gram of dry weight. Now, we'll come back to that. That's how much kilocalories, how much energy is in the plankton. Now, switch gears again. Now, working with the Motta people, we put satellite tags, pop-up satellite tags on these guys, And we want to know, so now we know the mouth area, we know the plankton density, we know the plankton density in the plankton toes per cubic meter of water, etc. But we want to know how much do they feed a day. So if we take the data from 4 sharks that we have good data in this area, and we analyze bin by bin, otherwise every minute of the day, we analyze these 4 sharks, and we're looking only at the depth between 0, which is the surface, and 1 meter. It's that accurate. It tells us that. You see, that is a plot of the percent time that these animals spend versus time of day on the surface, right? And we're presuming that when they're on the surface, they are surface feeding. That's the presumption. We're not sure, but when we see it at Holbox, every time they're on the surface, they are surface feeding. So you see, this is 4 o'clock in the morning, sunrise, they go up. They go up to the surface, and there's more time spent on the surface, like 60, 50-something % of the time up there is on the surface. At lunchtime, they go take a siesta. They go down below a little deeper, and then they pop back up in the afternoon, and by about 6 o'clock, sundown, they're gone back down. They spend less time at the surface. So this shows that for the SAT-TAG data, it's hard to see the black on here. When you take the bins times the percent time, it ends up being about 7.5 hours per day is spent for these 4 sharks between 0 and 1 meter. So we got an estimate of how much time per day they're now feeding on the surface. Then the guys from the Mexican group went up. They go up in ultralights, and they fly over them, plus in boats, and they look at the probability of sighting. So these data are actually quite biased, because what they do is they go up and they fly, and they typically fly in the morning and they come back in the afternoon. So it's tapering in the afternoon in part because they fly less in the afternoon, which is a little misleading. But this is the probability of sighting the sharks at different times of day. And there again, here it is, sunrise, lots of them, and then it's a little different, skewed a little bit to the right in this case. By noon, the probability, and dropping down again, and they stop flying in the afternoon. So we see the same peak. They go up in the morning, and then by about noon, they start coming down. I think it's related to temperature. I think the plankton is going deeper as later in the day, and the plankton is going deeper, and they're going down. So let's talk about the anatomy. We'll come back to all this in a second. This is a nice illustration by Emily Damstra. She's a biological illustrator, and it's a little inaccurate because I hope to redraw it, because she, of course, didn't know the exact anatomy. But you see the water comes in here. These are the filtering pads. They're pads like this. So you can see they're like this on one side, like this. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 20 pads like this, filtering pads in there. They're maybe derived from rakers, gill rakers. I don't think anybody knows. We're not sure. But the point is, if you go in there, stick your hand in there next time you're in there playing with the sharks, and put your fingers between them, you will see that there is actually no way for the water to go between them. They're sealed. It's sealed here, it's sealed here, it's sealed here, it's sealed here. The water has to pass through the pads. It cannot go between them. Like our gills, it can't go between the gills and go between them. The esophagus is back here, so it's truncated the esophagus. This is an example of the gill tissue. I'll come back to that too. So what does it actually look like up close? Well, this is from a moribund whale shark that we dissected. And you will see here on the pad, this is the left anterior pad, number 1, and you can see number 5. They're asymmetrical, slightly symmetrical. And what we've got, the upper pads are folded over. So this is the left side of the animal, so the mouth would be up here. That pad would fold over this way. This is the ventral here, 1, 2, 3, 4, 5, and that would be 1, 2, 3, 4, 5 on the upper, on the left side. That's like that. So you see the water must go through this. That's, I think, 6-inch ruler, right? So now, if we can see this, there is that pad we showed up there. Here is one of the pads taken out of it. So again, I set the task to the students. We take 9 areas on each one of the pads we had. We didn't have both sides from the whales, from the sharks that died, but we had most of the pads. So we take 9, and within that 9 areas, we also, I'll show you a blow up of this. This is what it looks like, the filtering mesh. I don't call it pores, it's a mesh. We sit there and we measure the diameter of all the little openings inside there and the area formed by every one of those pores. If you turn that pad upside down, this is what it looks like on the other side. There's a series of vents like this, and we can call them, there's primary veins here. This is the actual filtering pad here. There's these ridges we call primary veins, and another ridge called secondary veins, and that's the gill tissue. So the water and a whale shark has to go through here. The plankton gets trapped maybe on the surface. The water comes out through here, and then is directed over the gills and then comes out the gill slit. See? And you can see these veins are pointed towards the exterior. So this is the cross-section through one of the pads. So here is the actual filtering pores. These are the pores, the sieve-like mechanism. It looks like a sieve. It looks like a kitchen scouring pad. There. It doesn't look like anything. There's no other shark that has anything like this. These are the pores. See, these were the primary veins I was talking about, like this. They're like veins. These are the secondary veins. Now the water has to go through here, through these veins, out here like this, and up over the gill tissue before it exits the shark. This is important. That is the mesh. That's a close-up. That's what the mesh looks like. Very pretty, actually. See? They're not pores. They're irregular in shape. You are now looking through them, through those veins, the first primary and secondary veins, to the other side. So what we did is we measure all these little distances here and the area in there. In 9 areas in each pad, hundreds and thousands of areas, measurements. So here is a 623 centimeter total length shark. Here is a 631 centimeter total length shark. Just to give you an example. These are the sharks that died. And here is, so we're going to, red is one shark and blue is going to be another shark. So this is the mouth area. Based on all those measurements I've done, from the different size of the shark and so on, that is approximately the mouth area of this shark, and that's the mouth area of that shark. That is the filtering pad area. Total area, not holes, but the thing. You can see the filtering pad on this shark is 10,000 square centimeters, and the actual mouth is only 1,800 square centimeters. It's not even a square meter, right? So look at this one. Now, so the mean pore size, you measure the pores, those little pores on all the sieves, the pore size is 1.2 millimeter across the pores. Remember, fish eggs can be 0.6 to 0.8 millimeters. So fish eggs are smaller than the pores, right? So you then calculate what's called the open area ratio. So you have these pads. What of that pad, those 10,000 square centimeters, is actually open, is holes. And that is, the open area ratio is 53. That means 53% of that pad is open where water can flow through. So therefore, you have 4,000 and 6,000 square centimeters of open area that water can pass through. See? So you've gone from, that's the mouth of this shark, and that's how much area the pad is, much larger than the mouth. Of course, that makes sense. You wouldn't want a sieve that's smaller than your mouth. So, what is the average plankton length? The average plankton length varies from 1/8 of a millimeter to 10 millimeters. So here is the pore size, here is the plankton size. So most of the plankton would get stuck on the pores. Nelson and Eckert found that's the average plankton size, 1.5 by 0.5 millimeters. And the Belize Barrier Reef, they found fish eggs were smaller than the pores. So they can eat plankton that is smaller than the pores in their pores. So the question is, how do they trap the plankton? Well, I won't even try and do the math, right, because that's not my forte. So we send it off to University of California, and the computational fluid dynamics people over there then calculated, they use the pressure within the mouth cavity using Bernoulli's equation. Then they calculate all the measurements we give them. They calculate the pressure across the filtering pad, and then from that they calculate the flow velocity of the mouth. Otherwise, if you have a shark that's going 1.1 meters per second, it's swimming through the water, you cannot assume that water is entering that mouth at 1.1 meters per second. It's actually less, because the water hits the side of the mouth and so on, and there's resistance. And when you do the computational fluid dynamics, it shows it the flow is actually about 90% of the flow. See? So you want to know the volume moving into that mouth. It is 90% of the swim speed of the animal. It's not 100%. That's a mistake. There are some people, there's actually a thesis of feeding of whale sharks in Baja California, and they didn't do this. So there's a couple of papers out there, and they just assume that it's a tube, and when you ram the tube through the water, whatever the swim speed of the animal is the speed of water going in, and you'll get errors in your math that way. So now you sort of get ready for this now here. So you have the ram filter swim speed. So they swim at 1.1 meters per second. But the actual speed going into the mouth of water is 90% of that. So that is 0.99 meters per second. That's the flow of water going into the mouth, right? Okay, 4. So for the two, we got two size sharks. I got a little shark and a bigger shark to show you for comparison. If you 84.7% of the mouth is underwater, so you cannot take the whole area of the mouth. So the area of the mouth that's underwater, of their total mouth area, gives you the effective mouth area. That's 914 and 1659 square centimeters of mouth that is now plowing through the water. Open, right? Now we got the speed and we got the size. So the small shark ingests, how much water does go through the mouth? It's going to be this square, that's that, in terms of square meters, times the speed, meters per second, right? And you get meters times meters gives you cubic meters per second. That's how many cubic meters of water per second go through it. And if you multiply by 60 and multiply 60 again, they are filtering estimate, rough crude estimate, 300 cubic meters in an hour. That's how the sharks are plowing through the water. The big one is doing almost 600 cubic meters per hour. That's a lot of water. All right, now we know from you guys, your work, that the plankton biomass is 4.5 grams per cubic meter of water at the feeding sites. So you multiply this times that, and you get the small shark is ingesting 1.5 grams per hour of plankton, and the big guy is ingesting 2.660 grams per hour of plankton. They feed on average, roughly, very roughly, 7.5 hours per day. There's probably a lot of error in that. So you multiply that of grams per hour times the number of hours, right? That gives you how much grams per day. You found out how many kilocalories of energy is in 1 gram. So you multiply that, and ta-da, the small shark is eating, estimate, 3,565 kilocalories per day, and the big shark is feeding about 6,500 kilocalories per day, which seems really low. That's not even a Big Mac and a Whopper and cheese, right? They're subsisting on a Big Mac and a Whopper and French fries. That's it, per day. So that looks kind of suspicious, right? From you guys, the captive, from Leslie and Mike and those guys, the captive whale sharks here, your sharks, 515 centimeters and 664, roughly with the same size sharks, at your aquarium are feeding at, you're being fed 4,000 to 7,500 kilocalories today. That's what you guys are feeding them. So you are feeding them and they're gaining weight. Look at that versus that. There's our estimate, 35, 36, 64. And there's what you're feeding them per day, which kind of lends credence to it that, wow, this is a really relatively low, it seems, amount of kilocalories per day to subsist on, but your animals are seem to doing and gaining weight on that. If we were an order of magnitude off, I would worry, but. So I want to compare this to, there's stuff for basking sharks off England, so I want to compare that to look at the literature on the basking sharks. So that seems to answer some of that. So how are they filtering the plankton out? The bottom line is we don't know. But this is dead-end filtration, and this is called cross-flow filtration. This is from a paper by Beth Brainerd. You take a sieve in the sink, a strainer, and you pour water with plankton through it this way. That's called dead-end filtration. And it's not that simple, but actually your plankton can get trapped many ways. Your plankton can get stuck between the sieves. Your plankton can get stuck on the sieve, and there can be electrostatic attraction that can attract it, etc., etc. But you know this clogs up really, really quickly. In industry, in the beverage industry, they use this. This is called cross-flow filtration. Your fluid comes in and hits the sieve at an angle. Either your pad is at an angle or the water comes in at an angle. So what happens is the water goes through the sieve. The math is very hairy. I don't even begin to understand it. The water goes through the sieve, but most of the particles skim over the top of the sieve and collect in the rear of the sieve. That's what they use in the beverage industry because they don't have to clean the filter so often, right? And I think this is what the whale sharks are doing. I think the whale sharks, the lower pads in particular, are at an angle. So when the whale shark is swimming through the water is coming in, it's going through those pads. Remember, the area of the pads is much greater, the hole is much greater than the mouth area. The plankton is going to the back of the pharynx, right, by the esophagus, and I bet you if you look in there, one of you guys should crawl inside the shark when it's filter feeding and look for me, and there'd be a bolus of food swirling around in the back of the pharynx. We need to see that. And I bet you every now and then something triggers it, and they swallow that bolus, and then they keep going. But it will clog. The pad will clog every now and then, and the video didn't work. I forgot my video, but I have a beautiful video. The shark swims along, and we timed it. It's about 3 to 13 minutes. Every 3 to 13 minutes off Mexico, the shark comes to the surface, and he coughs. He goes, bleh, and a bolus of white stuff comes shooting out of his mouth. Other people have observed this too. Even when they're doing subsurface feeding, they do the same thing. That, to me, is the pad is clogging up to some extent, and something triggers it. The flow rate, something changes. They come up and they cough to clear the pads, and then they go right back down again and do it. So let's see if it turns. So lastly, the last thing now is, what is the significance of these veins here? Again, this is hypothesis. This is the hypothesis we have. The open area ratio of this, that is the open area ratio, the area of the holes to the total pad, is 0.53. If you design a flow tank, they use this in fluid dynamics. You design a flow tank and you want laminar flow, more or less laminar flow. Flow equals flow across the tank. What you do is, there's engineering papers on this. You put a mesh screen, and then over the outside of the mesh screen, you put what is called a collimator, which is like this, to get a laminar flow. The theoretical from the engineering calculations for the mesh screen is 0.57 is the optimum of the thing. That's the open area ratio of the shark. That's the optimum for laminar flow. The collimator length to diameter is, on the sharks, that is the width to the diameter of these channels here is 6.5, and in a flow tank, the optimum is 6 to 8. So I think what they're doing is, this is designed through evolution, so when the water goes through the pores, it comes through here, it forms more or less laminar flow over the gills with microturbulence. It would cause microturbulence, little teeny turbulence within laminar flow like that over the gills for maximum gas exchange over the gills. So that's just a hypothesis. And that's why fluids engineers would have to test that. So in Mexico, the whale sharks seek out dense patches of zooplankton, mostly shrimp, copepods, chaetognathes, and fish larvae. They ingest between 1,500 and 2,600 grams per hour of plankton. The filtering pads completely occlude the pharyngeal cavity and are about 6 times greater than the mouth area. The average pore size is about 1.2 millimeters, which is sometimes less than or greater than, depending on the plankton size, which ranges from 10.6 to 0.8 millimeters. The surface ram filter feeding, they approximately filter feed for about 7.5 hours per day. The sharks of about 4.8 to 6.3 meters of total length ingest about 3,600 to 6,500 kilocalories per day. Captive whale sharks your guys here ingest about the same amount for weight gain. And ram filtering may involve what's called crossflow filtration, concentrating the plankton in the posterior pharynx and reducing the clogging. And then the open area ratio and the collimator size matches theoretical predictions for microturbulence generation across the gills. And I think that is just to thank you, Georgia Aquarium, University of South Florida, Motta Marine Lab, the people in Mexico, and NOAA, etc., for all this support through the research, for both Motta and our research.